I've made a lot of videos so far that talk about how we can create diverse antigen receptors. And this is kind of another in line with those. There are a couple ground rules for making a diverse set of antigen receptors. And they're all genetic mechanisms. And thankfully, they are shared between the TCR and the BCR. I'm not going to go into all of the processes. This is a little more of an overview video that will kind of go through some of the tricks that the body uses. So each T or B cell makes an antigen receptor that is specific for one antigen and one antigen only. It doesn't matter if there's, one BCR on the surface or 50 million. All of them will have the exact same antigen specificity. And that's because it is controlled at the genetic level as to what is actually expressed. But let's think about what this actually means. If each cell and T cell can only recognize one epitope of one antigen, you would need to make, say, over 100 billion B cells to make antibodies and TCRs to recognize the at least 100 billion antigens that you could potentially come in contact with over the course of your life. Well, that's a really big problem. In the context that we don't have enough genetic material to do that. Now, the good news is we're actually able to do this. You have this. Um, in fact, every lymphoid tissue of your body, you probably have at least 10 billion unique receptor cells. So there is enough variability, but it's not based in our genetic code. Our genetic code actually has very little to do with this. It has everything to do with being creative. We don't have a separate gene for each antigen specificity. That's not possible. They sequenced the human genome almost 20 years ago, and the total genes necessary to create a human is only about 25 to 35K. So there aren't enough genes to encode for the amount of antigen receptors we need. So the trick is creating a system that allows for significant variability in the amino acid sequence that the receptors can reorganize while still creating a functional antigen receptor. So we've got a couple ground rules for how this could work. So first off, we have a couple goals. You need to make a massive amount of diverse antigen receptors. And remember, we only have 25 to 35,000 genes for the whole human. So we've got limited resources. Also, we can't spend all of our energy making antigen receptors. While I love immunology, the entire body still needs to function. And I think your physiology professors and your neuro professors might argue that the brain and heart are just as important. So we can't use all of the energy. Also, whatever we make is going to be super potent. So we don't want it to attack the host. So let's set up a couple ground rules that we can all agree with, and then let's come up with a mechanism to create this incredibly diverse population of antigen receptors that we need. So rule one cell, one antigen specificity. We already talked about that. Second rule, every single antigen receptor in your body was created before you ever saw a single foreign antigen. Think about that. That's mind-blowing. This isn't done reactionarily. This is done preventatively. It's like the initial preventative medicine. It's done before you're ever exposed to them. And no, they aren't necessarily passed down from year to year or generation to generation. All right, next one. Because we have to make so many, if we think of our lymph node like a bag of marbles, we're only going to have like a couple of each type of antigen specific cell. So if each of these dots of color is a different antigen receptor, I've only got one. But let's say the pathogen I'm fighting right now is orange. That's going to be the one I'm going to clonally expand so that I can make more and more and more of these until I fight off the pathogen I'm trying to get at. Okay? All right, so we're going to clonally expand the one cell that can actually help us in the fight. Whenever possible, we're going to make things cross-reactive. It's going to limit the amount of cells we need to make. Okay, so what does cross-reactivity actually mean? Let's say we make 100 billion antigen receptors, okay? Now, that should get it, right? But what happens when the 100 billion and 1th antigen comes along? Well, there are only so many conformations that peptides, lipids, carbohydrates, and other organic materials can take on that will have the right hydrophobicity to be identified as an antigen by an antigen receptor. So even though each receptor is specific for only one antigen, that one antigen might be shared by several different pathogens, okay? So it's possible that it could be used against several different organisms or compounds, and that's the case of cross-reactivity. All right, number 5, we're going to put all of the genes for the TCR and BCR on different autosomal chromosomes. Okay, so you don't want to knock out one chromosome and destroy the whole system. So remember, the B cell receptor has a heavy chain and a light chain. The T cell receptor has a beta chain and an alpha chain. Okay, and then within that, you've got V, D, and J here. You've got VJ kappa and VJ lambda. You've got V, D, and J here, and you've got VJ here. Okay, so these are all of the things. So all of this one is going to be on one chromosome, all of this one on a second, all of this one on a third, all of this one on a fourth. And that way we don't lose the entire system if, let's say, they were all on chromosome 1 and we lost chromosome 1. We still have some backup plans, all right? It's important to note that these are all autosomal chromosomes. They are not sex-linked chromosomes, so you will inherit and use them no matter what. Okay, the last and most important thing, only rearranged genetic material gets made. So remember, you've got all of these V regions, D regions, J regions. We copy them, we keep them, but we don't make any protein with them until they are rearranged, because those are the ones that are actually functional antigen receptors. They're not a functional antigen receptor until they've rearranged, and that's key. All right, I keep saying this word rearranged. What do I even mean by rearranged? So rearranged actually has to do with the VDJ regions themselves, okay? So if this is a gene line, right, and I've got all of these V regions and all of these D regions and all of these J regions, right? What eventually I want to have happen to make a beta chain or a heavy chain is I want one V region, one D region, and one J region all together. So let's say I took this one and this one and this one. And that's going to be different than if I had taken a different set of them, like that one, and that one, okay? So that's a VDJ rearrangement. It's just the careful way in which the genes that encode for the antigen recognition region of the receptor are spliced together. I want you to think of your V, D, and J exons just as Legos. And just like if I gave you a bucket of Legos, could you build a different shape? Of course you could. Or if I gave you like a bunch of train cars, you could put them in a different order. Who says the caboose has to be in the back and the engine has to be in the front? Nobody. It's up to you, okay? And that's pretty much how your body feels about V, D, and J. Now, there is a little bit of an order. Sorry, the engine probably should be in the front and the caboose probably should be in the back. But the rest of it we can pretty much make up as we go along. So there are about 5 mechanisms that we're going to talk about that get us through making this diverse method, okay? Mechanisms 1 through 4 take place in both B cells and T cells. Mechanism 5 is somatic hypermutation, and this is a superpower that only B cells are capable of. And we'll talk about it more when we talk about activating B cells in a later case. All right, the first mechanism of diversity is something you've probably already figured out. We inherit a ton of V regions. Lots and lots of V regions, okay? So you already know that each BCR and TCR contains V domains. These V domains on either the heavy or the kappa and lambda or alpha and beta chains, whatever, are encoded by V gene segments, okay? And these V gene segments are actually what are found in our germline DNA, okay? So in the germline DNA is where we're going to find the V gene segments. We inherit multiple V region gene segments. The gene segments are split into separate exons, okay? And these exons are actually what are going to be put together, spliced together in order to make our V region. So basically, you can use the same constant region if we're talking about a T cell receptor or even a B cell, right? So for a TCR, it would be like your alpha or beta chain constant region. When we're first creating B cells, it's always mu, right? That's your IgM, that's always first. So you can put that together with many different V domain genes. Each V region has a leader sequence followed by 95 amino acid coding sequence that basically tells you where to cut, okay, where you're going to make your cuts to get your various gene exons that you want. For antibody molecules, there are about 100 of these germline V region gene sets for each of the heavy chains and fewer for the kappa and lambda ones. During development of a B cell, one of these V regions is chosen at random from the heavy chain set and one from a light chain set. While this mechanism generates a significant number of different combinations and therefore a large number of different antibody specificities, it's not enough to account for all the specificities that each person produces. So we're going to try to recombine this using different V, D, and J regions like I've shown before. All right, so this moves us into the second mechanism, recombination of the gene segments. So this literally just means that any V exon can be used with any J exon. And if we're talking about a heavy or a beta chain, any J exon can be used with any D exon, and so on and so forth. Each V chain encodes for 95 amino acids, right? So if I've got a V chain, and a V chain is going to have a V, a D, and a J, okay? Together, these three should be 95. But the actual weight is about 110. So where do the other 15 amino acids come from? And that's the J sequence, okay? So the joining. The J sequence joins the V region to the constant region. So then it goes on, here's your constant, and we need to join these together, okay? And you can do this in a variety of ways. Any V exon can be used with any J exon to create more diversity. So in a kappa light chain, you have 35 V regions and 5 J regions and 1 constant region, which is your kappa light chain, okay? So theoretically, you could have like V3 with J2, which would have a different specificity than V3 with J1, than V4 with J2. See what I'm saying? All of these are slightly different. And all we did was just reuse different pieces. And now we have a different light chain, which gives a different antigen specificity. The same thing goes for the heavy chain. And as I mentioned before, only the recombined DNA is going to be expressed as a protein. And the heavy and beta chains always do this process first. All right, if you're following along in the notes, I just jumped ahead to the third mechanism of diversity. Don't worry about it. Go back over those 2 paragraphs. I did quote 2 videos by Armando Hasadunagan, who I actually think does a really great job on antibody somatic VDJ recombination. I'm not going to ask you about that in detail. I just want you to know it exists, that basically they're going to recombine. Now, if at this point you're like, oh man, I have no clue what you're saying, this is all so confusing. Don't worry, you're not alone. The T cell is confused too. It's so confused that it's going to start making mistakes. Accidents happen. And in this case, they're a really good thing, because the next mechanism of diversity is recombinational or recombinatorial inaccuracy. Basically, it's trying to put all of these exons together in such a quick pace that it's going to make some mistakes. And this is the one time where making a mistake during genetic recombination is a good thing, because mistakes increase variability. And we want variability here, right? Mistakes are wonderful here. So while the heavy chain variable regions are undergoing recombination of first D to J and then V to DJ, all right, there's going to be an enzyme activated. This enzyme is known as TDT for terminal deoxyribonucleotide transferase. That's a mouthful, so let's just go with TDT. Basically, this enzyme inserts nucleotides between, okay? So if I had a V, a D, and a J, I could have V plus G, R, A, and these are amino acids, or just A, or just G, or whatever, right? And in doing so, in adding any nucleotides, all right, not amino acids, nucleotides. So let's just change these out for some C, T's, G's, and A's, okay? By adding in nucleotides, I have changed the amino acid signature. When you change the amino acid signature, you also change the protein that's made. And remember, only recombined proteins are made. So we just changed what this would be by adding any nucleotides, and that's why they're called N-nucleotides. This creates additional diversity in the messenger RNA that's transcribed from this DNA. TDT is not active when the light chains rearrange, so the mechanism generates variability only in heavy chains. But you can still have recombinational inaccuracy just at splice junctions. So anytime we have two genes coming together, there's likely to be mistakes. So with one or two base pairs beyond those that encode amino acids being utilized or affected. All right, we're on to diversity mechanism number 4. Now remember, when we were making our T cells and B cells, in the TCR, we made the beta chain first. In the BCR, we made the heavy chain first. In both cases, it was paired with kind of a surrogate, like a training wheels extra chain, right? So for T cells, it was the pre-T alpha. And for heavy chain, it was the surrogate light chain, okay? And these are just training wheels, right? They're not an actual alpha chain or light chain. They're just to see if the beta chain or heavy chain we made can actually be functional. When that happens, remember that the cell is going to proliferate wildly. We're going to get tons and tons of these cells. And these cells only have, at this point, a beta chain if it's a T cell receptor that's being formed, or a heavy chain. We haven't made that second chain, but that's great. That's so great. You know why? Because this beta chain could be paired with this alpha chain when it's made, or this alpha chain, or this alpha chain, or this alpha chain. You get the picture, right? So we took the same beta chain and we paired it with a multitude of different alpha chains. And that right there, my friends, upped the diversity so much. So this is huge. It's waste not, want not. It's called random assortment. It's random assortment of chains. Any beta chain pairs with any made alpha chain. Any heavy chain pairs with any made light chain. All right, last mechanism of diversity, and we're not even really going to go into it here. This is known as somatic hypermutation. I have another video on my page that goes all into this that you guys can watch. I'll cue you for that when we're ready for it. But really, this is something only B cells do, and it increases their ability to be really good antigen binders. As B cells divide rapidly in germinal centers of the lymph node, there are a lot of opportunities for mutation. And while these mutations could be detrimental to individual B cells, these B cells, the ones that lose some specificity, will be eliminated, and the ones that gain increased affinity will be proliferated and maybe even turned into memory cells. So it basically just increases the quality of the antibody or B cell response. Okay, not so much the quantity, but the quality. It refines it. Like I said, we'll go through that in a later video in a later case.